Hello everyone, welcome back to Believers Science Academy, Bangalore. This is Prelims Hacks topic of the day series. In this, I will be discussing a short but very significant topic from UPSC Prelims 2025. The topic is recombinant proteins. What are recombinant proteins? They are the proteins artificially produced by introducing specific genes or the specific genes in a recombinant DNA into the host organisms like bacteria or yeast, directing them to manufacture the desired proteins. Okay, so this is the basic criteria here. Normally, how a protein is produced is there is DNA, and then this DNA is converted into RNA, from RNA to protein, from protein to amino acids. Okay, so this process is called as transcription, meaning here all the sequence of DNA, ATGC, etc. sequence will be transcripted, and then they will be translated to form the protein. From protein, they will be folded to form amino acids. Okay, so from DNA, protein is formed. Okay, but in recombinant DNA, a recombinant protein practice, what happens here is the DNA will be modified by inserting a particular gene. That DNA which has been inserted with a new gene is called as recombinant DNA. Then this recombinant DNA is infused into a vector or a host organism such as yeast, bacteria, etc. Once they are included, that bacteria or that yeast starts to produce the desired protein according to the recombinant DNA. Then they are cultured in the labs to produce in a significant amount, and they are infused into human beings for the medicinal purposes. That is the simple process under recombinant DNA. Now, recombinant proteins. Types of recombinant proteins are interferons, recombinant hormones, tumor necrosis factors. Commonly are used in biomedical and therapeutic applications. Best examples are human insulin for diabetic treatment. Generally, insulin is produced within human bodies, but in certain patients, the capacity to produce the insulin is very less in insulin deficiency human beings. For them, this insulin is grown outside in the lab through the culturing of insulin, and then that is infused into the human bodies. Okay, that is the use of human insulin as a recombinant protein. There is another method where CRISPR-Cas9 technology is used. What they will do is the gene or the defunct gene in the human body, defunct in the sense the gene which is stopped working. The gene is supposed to produce the insulin, now it has stopped working, so that is called as defunct gene. That defunct gene will be removed using the molecular scissors, and the functional gene will be replaced in the DNA, and then the body itself starts producing the insulins. That is the advanced step which comes under CRISPR-Cas9. That is different, this is different. Okay, other example is growth factors for growth hormone deficiencies. Growth factors are also proteins, recombinant proteins produced outside and infused. And then factor 8 for hemophilia treatment, and therapeutic monoclonal antibodies for cancer and viral infections such as SARS-CoV-2. Okay, these are some of the examples. And the production process which I already discussed, one is you are going to insert the gene, that is the modified gene is inserted into a vector, then they are transferred to the cells, the vector is introduced into the host cells, then the cell growth, host cells are cultured for a high protein concentration, then there is a harvesting, that cells are harvested to obtain the recombinant protein, and after the harvesting, these recombinant proteins are infused into the human beings for medicinal or therapeutic purposes. Okay, applications are for biomedical research to study health and disease mechanisms, biotherapeutics, in drug development and disease treatments are also the recombinant protein is used. Then protein based polymers, these are used in drug delivery systems. Then disease treatment, where produces enzymes and antibodies for treatment. Then tissue engineering, provides scaffolds for tissue growth. Okay, these are the various applications of monoclonal and recombinant proteins. Now, what is protein? Okay, what is the protein? It is not the protein you take when you go to the gym. It is talking about the proteins that is produced in the human bodies. Proteins are essential molecules in biological systems, facilitating processes like gene expression, cell growth, and intercellular communication. Proteins are the basic building blocks of all kinds of processes in a human body. They are necessary for expression of a particular gene. They are necessary for a growth of the human beings. They are necessary for the intercellular communications. There are various proteins which helps in triggering of hormones, which in turn help in the growth and other activities. Then they provide blueprint for synthesis. DNA provides instructions for the synthesizing proteins through messenger RNA, which I've already explained about the transcription, translation, and the folding process. So this is a short topic about recombinant DNA or recombinant proteins. Very important. Don't confuse recombinant protein with CRISPR-Cas9 technologies. They are almost similar but very significantly different. Okay. So this is a short topic. Hope you like the video. If you like the video, then please click on the like button, comment on the video, and also share with your friends. With this, I will end the video. Jai Hind.